So, good morning to all of you first semester students. Today, I'm going to discuss with you about a cell biology topic that is given in your syllabus. That is about the endoplasmic reticulum, and we will actually discuss about the structure and the role in cells. What does endoplasmic reticulum do, and its types also. So, let's get started now. So, about the endoplasmic reticulum, they are a part of endomembrane system. So, what it is written here that I have given in your notes, the endoplasmic reticulum is the main component of the endomembrane system. Okay, it is the main component of the endomembrane system. Endomembrane system is also known as cytoplasmic vacuolar system. It is also known as cytoplasmic vacuolar system or cytocavity network. Why it is written cavity? Cavity means the space region, the hollow regions. Okay. Now this endomembrane system, as I have said, that this endoplasmic reticulum is the component of endomembrane system. This system mainly comprises of the nuclear envelope. It also consists of the endoplasmic reticulum and the Golgi apparatus. So, three important uh, components are present in the endomembrane system. The nuclear envelope. When you will study about the nucleus, you will see the nuclear envelope that consists of two non-identical membranes. So there are two non-identical membranes found within the nuclear membrane, one opposed to the nuclear chromatin and the other separated from the first membrane by a perinuclear space. So two membranes will be found, and inside there will be a space, and that space is called the perinuclear space. Perinuclear space. Okay, so that is your nuclear envelope and form a cisternae like structure, just like we have seen in just like we will see in mitochondria. So nuclear envelope is also a component of endomembrane system. It is composed of inner membrane and outer membrane, and in between there is a space called perinuclear space. The endoplasmic reticulum is also a part of endomembrane system, and the Golgi apparatus, which is mainly related to some terminal process of cell secretion. Now, those which proteins are to be secreted out from the cell are called secretory proteins. Those proteins has to be packaged inside the Golgi apparatus. Secondly, inside the Golgi apparatus, further modification of the protein structure occur, like glycosylation, in order to be uh, stabilize its structure and to be transported to the outside the cell. Okay, so the GERL, where we can say M as GERL, where G refers to the Golgi apparatus, ER refers to the endoplasmic reticulum, and L refers to the lysosome. They also forms a special region of the endomembrane system which is more related with the Golgi apparatus and involved in the formation of lysosomes. Well, lysosomes is a structure found inside the cell which contains various enzymes, various hydrolytic enzymes. It has acidic pH, and for the formation of lysosomes, your Golgi apparatus is involved. Okay, that forms the GERL system. And on the other hand, the endomembrane system, where transport of the proteins, um, the modified proteins occur from your endoplasmic reticulum to the Golgi apparatus, including your nuclear envelope is also involved here. Okay, that forms the endomembrane system. Okay, so now let us see this main feature of this endoplasmic reticulum. Generally, it is saying that the entire endoplasmic reticulum has two components, have two phases. One is the cytoplasmic or protoplasmic phase, and another one is called luminal phase. So, endoplasmic reticulum have two. It has a barrier which separate the cytoplasmic components, and if you see the membrane of this endoplasmic reticulum, or if you see the membrane of this endo membrane system, it consists of two sides. One is called cytoplasmic side or protoplasmic side. You can also say the cytoplasmic phase or protoplasmic phase, and another is called the luminal phase. The luminal phase is that space, is the phase where it actually, that membrane actually is located near the lumen, the hollow space. Okay, where the proteins are to be inserted into that luminal phase, and they will be post translationally modified. The proteins are to be modified, like glycosylation means addition of carbohydrate to certain regions of that proteins in order to stable the protein functions and structure. Okay, so if you see the figure here, you can see the luminal phase and the cytoplasmic phase are being observed. So this is the cell and these are the cytoplasmic part. Well, these are the endoplasmic reticulum. I'm sure you know about the rough and the smooth endoplasmic reticulum. You have studied in your junior classes as well as in your class 11, 12. Well, rough endoplasmic reticulum are the sites where ribosomes are attached to the membranes of the endoplasmic reticulum. So these are the ribosomes, which are the sites for protein synthesis. Okay, so they are located on the cytoplasmic phase or protoplasmic phase of the ER membrane, whereas the luminal phase is this one. This is the lumen. The green one is your lumen, the empty space, where the proteins which are synthesized from the ribosomes are to be entered. Okay, so if this is the ribosome, then the proteins, as I will show you in orange color, are synthesized inside the inside the lumen of the endoplasmic reticulum, and they are inserted into the endoplasmic reticulum inside, where these proteins are actually goes and they are further glycosylated for their stable protein conformation structure, and then they will, if it is a secretory protein, they will be now, with the help of secretory vesicles, they will be moving along towards the Golgi apparatus. They will move along the Golgi apparatus with the help of these secretory vesicles contain your protein part, and further within the Golgi apparatus they are further packaged, they are further glycosylated, means more addition of carbohydrate molecules attached for them to proper function, so that the proteins may function in their secretary path, in their secret, in their role. Okay, and for stability of the structure also, and then they are finally pinch out with the help of the secretory vesicle containing your desirable proteins, secretory proteins. So this is your, if I say this is your cytoplasmic phase and membrane, the membrane which is facing towards the cytoplasm side, this is your cytoplasm, and this membrane is located with the cytoplasmic, it is facing towards the cytoplasm, so it is cytoplasmic phase, whereas the black one that you can see here, it is a luminal phase. Well, luminal phase is that region of the plasma membrane which is facing towards the lumen, and this is your lumen, the green part, the empty space where the secretory proteins are to be glycosylated, they are further modified, and they are transported towards the Golgi apparatus for further packaging and for further glycosylation. Okay, so this was about the general membrane view of your endoplasmic reticulum. And if I see the morphology, if I see the morphological structure of endoplasmic reticulum, morphologically, the endoplasmic reticulum may consist of the following three forms. So there are three forms of your endoplasmic reticulum. One is your lamellar form. Lamellar form, also called as cisternaiform. Well, cisternaiform, or you can also say cisternai, a closed fluid filled sac, vesicle or cavity is called cisternae. So if this is a sac like structure which composed of two membranes, well, the endomembrane system always contain the two membranes, whether you are seeing the endoplasmic reticulum or Golgi apparatus, or whether you are seeing the nuclear envelope, they composed of two membrane, inner membrane and outer membrane. So endoplasmic reticulum can be observed in the form of cisternae structure, or you can say cisternae structure, which is a actually a vesicle or cavity where inside there may be some fluid present or may not be present. Okay, so that form of endoplasmic reticulum can be observed. Second form is the vesicular form, or simply a vesicle. And the last form that we normally draw in our diagram when we are drawing about endoplasmic reticulum is the tubular form or tubules. So in this picture, you can see the two types of endoplasmic reticulum, the smooth endoplasmic reticulum and the rough endoplasmic reticulum, where ribosomes are attached to the surface of the cytoplasmic surface of the ER membrane. And you can also see some you cannot see the tubular form. Yes, we can see the tubular form. These are the tubular form actually, and the vesicle form are also observed, which is pinching out here. Okay, let's see that structures. Well, uh, this is your structure. This is your different forms of endoplasmic reticulum: the cisternaiform, the vesicle form, and the tubule form. Well, if you see the cisternaiform, it is saying that they are long, flattened, sac-like, unbranched tubules which have the diameter of 40 to 50 micrometer. They may be small, that I have drawn with you, just like vesicles, but they will be separated. Okay, they will not be linked together, just like as we draw uh, in case of when we are drawing the endoplasmic reticulum structure. So cisternae are long, flattened, sac-like, unbranched tubules. They are not branched. They have no sub branches, and they have the diameter of about 40 to 50 micrometer, and they remain arranged parallelly. If you see the structure of your cisternae structure, they remain arranged parallelly. That means they are not interlinked. They are not interlinked. Okay, they are parallelly oriented in the form of bundles. Rough endoplasmic reticulum usually exist in cisternae form, which occur on those cells which have synthetic roles as the cells of pancreas, the cells of notochord, and brain. So normally cisternae form are not branched, they are unbranched sac-like structure, long flattened sac-like structure, as you can see in this picture, which have a diameter of about 40 to 50 micrometer, and they remain arranged in bundles. Okay, and these tubules are not interlinked, they are parallel with each other. And this type of endoplasmic type of form of endoplasmic reticulum found in RER, which is your rough endoplasmic reticulum, who and it is found in the cells which have synthetic roles, like the cells of pancreas, the cells of notochord, and brain. Second form that we can see is the vesicle form. Well, this type of form we can see in the picture in the previous diagram. The vesicles are oval in shape and they are membrane bound vacuolar structures having a diameter of 25 to 500 micrometer, and these vesicle form are often isolated from in the cytoplasm. They are not joined and occur in most cells, but especially abundant in the smooth endoplasmic reticulum. That means vesicular form mostly observed in case of ACR, that is smooth endoplasmic reticulum. Well, you can see different granule-like structures are observed on the rough endoplasmic reticulum in this cisternae. These are actually ribosomes, and I have said that most of the cisternae form of endoplasmic reticulum structure observed on rough endoplasmic reticulum where ribosomes are attached. The last form is your tubular form. Well, tubular form is mostly with what we draw in when we draw endoplasmic reticulum. Tubular form have a branch structures. As you can see, the branch are being observed here and form a reticular system along with cisternae and vacuoles. Well, the picture that I have shown before is of tubular form. It has vesicles, it has the cisternae like structure, and it has the tubules also. Okay, so the tubular form of structure is branched, it has branch. Okay, secondly, it is formed along with the cisternae and tubular form and vesicle form of types of structure that is found in endoplasmic reticulum. Diameter of the tubular form is about 50 to 119 micrometer, and they almost occur in cells. So the tubular form is the most common form, which is made up of cisternae form also, vesicular form also, right? Tubular form of endoplasmic reticulum found in smooth endoplasmic reticulum and are often dynamic in nature. That means it is constantly changing. It is associated with the membrane movements. What type of movement the membrane of the endoplasmic reticulum do, and then fission, that means separation of the membranes when the vesicles, secretory vesicles, pinch out from the membranes of the ER, and fusion between the membranes of your network between the Golgi apparatus with the ER, that can cause the structure of the tubular form to be dynamic. That means they are constantly changing depending on how the membrane is moving when they are pinching out from the cell, so that the pinching out from the cell, sorry, not pinching out from the cell, pinching out from the endoplasmic reticulum. Okay, for further processing of the secretory molecules into the Golgi apparatus, right? They have to go from here to here with the help of secretory vesicles, and that secretory vesicles is formed of that membrane, where we have to change the structure of the membrane at that time. So it is always dynamic, it is always changing. Okay, next is your ultrastructure. If you see the ultrastructure of the membrane, we will see what thing we will see. Let us see. It is saying that the cavities of the cisternae, vesicles, and the tubules of endoplasmic reticulum are bounded by a thin membrane of 50 to 60 angstrom thickness. So the membranes of um, if you see the cavity, the hollow space region, the hollow space region is called cavity. If you see the cavity region of the cisternae, vesicle, and tubular type of structure of ER, it is observing that they are also bounded by a thin, very thin membrane of 50 to 60 angstrom thickness. Okay, and the whole membrane of the endoplasmic reticulum is a fluid mosaic like structure, just like the fluid mosaic like structure that we found in the plasma membrane, that we found in the nucleus, as well as in the Golgi apparatus. So, the membrane which is found in the endoplasmic reticulum, both the inner and outer, they are fluid mosaic like structure, just like we see in the plasma membrane. Thus, it is a bimolecular layer of phospholipids which floats, in which the proteins are floats in various sorts. So that's why it is written. So the membrane thus composed of a lipid bilayer, phospholipid bimolecular layer which is constantly floating, and the proteins are floating in various sorts. Then it is written that the membrane of the endoplasmic reticulum is continuous with the plasma membrane and nuclear membrane and Golgi apparatus. Remember always, that's why they form a network of endomembrane system. The membrane of endoplasmic reticulum forms a continuous network with the membranes of the plasma membrane, with the nuclear membrane, and with the Golgi apparatus. So the membrane which is found in the endoplasmic reticulum, they are continuing with the plasma membrane, they are continuing with the Golgi apparatus membrane, and also with the nuclear membrane. Now, what is the importance of this empty region which is called as cavity, which is found inside the lumen of the endoplasmic reticulum, is the cavity of the endoplasmic reticulum is destined for passage for the secretory products, whatever secretory products. It may be a secretory protein or some waste products which has to be eliminated out from the cell. So normally, generally, we will speak here about the passage for the secretory products. So those products which are needed for the other cells to go around, okay, that has to be packaged here, that has to be formed here and packaged here, and it has to be transported to that destined cells via the blood vessels. So the cavity of the endoplasmic reticulum, the empty space which is found in the endoplasmic reticulum, acts as a passage for the secretory products. Okay, I think you have understood what I have said. So up to this much about the ultrastructure. I will not go further in this paragraph. Now let us discuss about the types of endoplasmic reticulum. Well, there are two types of endoplasmic reticulum. One is called smooth endoplasmic reticulum, also called as agranular. Agranular means no granules are present. And another one is called rough endoplasmic reticulum. Now, what it says about the smooth endoplasmic reticulum, this type of endoplasmic reticulum possesses smooth walls because ribosomes are not attached with the membrane. I think this line you understood very well. Here, ribosomes are not found with the attached with the membranes of the smooth endoplasmic reticulum. The smooth endoplasmic reticulum occurs in most cells which are involved in the metabolism of lipids, including steroids, and glycogen. Generally, smooth endoplasmic reticulum are the sites are found on those cells which are mainly involved in metabolism of lipids, like uh, including the steroids also, uh, where you can find this type of smooth endoplasmic reticulum, and glycogen, as you will see in the function that how they are involved in metabolism of glycogen for converting glycogen into glucose to get energy for the cell. So they are mainly involved in metabolism of lipids including steroids and glycogen. Secondly, the smooth endoplasmic reticulum is generally found in adipose tissue, in adipose cells, interstitial cells, glycogen storing cells of the liver, conduction of the fibers in the heart, spermatocytes, and leukocytes. So, where these smooth endoplasmic reticulum are found? Smooth endoplasmic reticulum are found in the adipose cells, in the interstitial cells, in the glycogen storing cells of liver, conduction of fibers of the heart, spermatocytes, and leukocytes, which are the type of white blood cells. Muscle cells are also very rich in smooth endoplasmic reticulum. Well, I have discussed about the sarcoplasmic reticulum in the plasma membrane chapter, where you have seen that how muscle cells are contract and relaxed by storing and releasing the calcium ions within the lumen of the endoplasmic reticulum. So the muscle cells are also rich in smooth endoplasmic reticulum, and the endoplasmic reticulum which are found in the muscle cells are called as sarcoplasmic reticulum. Okay. In the pigmented retinal cells, this endoplasmic reticulum exists in the form of tightly packaged vesicles and tubes known as myeloid bodies. Now, this endoplasmic reticulum is also found in pigmented retinal cells, and within the pigmented retinal cells, this endoplasmic reticulum exists in the form of tightly packed vesicles and tubes, and that are known as myeloid bodies, known as myeloid bodies. So many things has been revealed here when you are saying about the smooth endoplasmic reticulum. The first thing that we can see that this smooth endoplasmic reticulum occur in those cells which are involved in metabolism of lipids, glycogen, and including steroids, as you will see in the function. And this smooth endoplasmic reticulum found on adipose cells, in interstitial cells, glycogen storing cells of liver. It occurs also on the conduction fibers of heart, spermatocytes, and leukocytes. Muscle cells are also found very rich in this type of smooth endoplasmic reticulum, and that's why and they actually have this smooth endoplasmic reticulum in order for contraction and relaxation of the muscle cells. And here the name of endoplasmic reticulum, the name of smooth endoplasmic reticulum in the muscle cells are known as sarcoplasmic reticulum, where they are storing the calcium ions and they are removing out the calcium ions with the help of calcium channels. Okay, that causes relaxation and contraction of the muscles. Now, within the pigmented retinal cells also, this endoplasmic reticulum occurs, and they occur in the form of tightly packed vesicles and tubes known as myeloid bodies. Next, that we will see more about the smooth endoplasmic reticulum. Now, this region that we are calling as glycosomes, as you can see, I am zooming it in. So, Okay. Okay. So glycosomes are dense particles which are found in the membrane of smooth endoplasmic reticulum in the liver cells. Glycosomes are consist of enzymes and they involve in synthesis of glycogen. So what are glycosomes? As the name suggests, glycosomes, where they are the dense particles found within the membranes of smooth endoplasmic reticulum, especially in the liver cells. And these glycosomes contain enzymes which are involved in your synthesis of glycogen. Now, although this smooth endoplasmic reticulum, it forms a continuous system with the rough endoplasmic reticulum, it has different morphology. Now, when we see the smooth endoplasmic reticulum and rough endoplasmic reticulum, they form a continuous network with the rough endoplasmic reticulum, but the morphology is fully different. Okay, the morphological structure is different from the rough endoplasmic reticulum. Although they form a continuous network, the ACR, that is smooth endoplasmic reticulum, forms a continuous network with the rough endoplasmic reticulum, but still they have a different morphology. Like what I have said about the glycosomes, that glycosomes that is found within the liver cells, okay, which are the found in the liver cells, and they are dense particles which are found in the membranes of the smooth endoplasmic reticulum, and they are involved in the synthesis of glycogen. Okay, now this is only found within the smooth endoplasmic reticulum of the liver cells, not in the rough endoplasmic reticulum, although they form a continuous network. So there may be occurring that some portion of the glycosomes may occur in the rough endoplasmic reticulum, but it does not occur in the liver cells. It is only found in the liver cells within the smooth endoplasmic reticulum. Okay, that's why they have said that although smooth endoplasmic reticulum forms a continuous network with the rough endoplasmic reticulum, but still the morphology, it has a different morphology. Each of the endoplasmic reticulum has a different morphology. Okay, so this is the what they want to say actually. Okay, now let us see about the granular or rough endoplasmic reticulum. Well, as the name suggests, that the granular or rough endoplasmic reticulum possesses a rough walls because ribosomes remain attached with its membranes of endoplasmic reticulum. So, rough endoplasmic reticulum get its name because ribosomes are being attached in the cell membranes of this endoplasmic reticulum. Here, ribosomes play a very vital role in the process of protein synthesis, as you have studied in your class 12 also, that ribosomes are the sites where protein synthesis occurs, and the process where the protein synthesis is being occur by using the mRNA as a template, uh, also called as messenger RNA as a template, known as translation. Okay, the granular or rough type of endoplasmic reticulum is found abundantly in those cells which are active in protein synthesis, like pancreatic cells, plasma cells, goblet cells, and liver cells. So, this granular or rough type of endoplasmic reticulum, where it is found mostly, those who are continuously involved in protein synthesis, those who are which are continuously involved in those protein synthesis which has to be secreted out from the cell, right? Better to say. So, like for example, granular or rough type of endoplasmic reticulum found in where It is found in the pancreatic cells. It is found in the plasma cells, goblet cells, liver cells. Well, plasma cells is special type of what? B cells. Remember, B cells, B lymphocytes, which are the part of your immune system, cell mediated immunity. They secrete antibodies, which will be specific for particular antigen, right? So those cells which are actually continuously synthesizing, which are active in protein synthesis, they have this rough type of endoplasmic reticulum, like pancreatic cells, plasma cells, goblet cells, liver cells. Next, it is saying that the granular type of endoplasmic reticulum takes the basophilic stain due to its RNA content of ribosomes. Well, they take a special type of stain when you are doing laboratory staining to observe the rough endoplasmic reticulum. They stain the basophilic stain. Basophilic means that have base, actually basic in nature. Two type of stain are there: acidic stain and base stain, like acetocarmine, acetoarsine. These stains are acidic in nature, where basophilic stain, they are basic, and these stains will be stained by the rough endoplasmic reticulum because they have ribosomes attached now. That's why they will stain. Okay, now I'm not saying this part from here because we are discussing about the rough endoplasmic reticulum structures and functions. Now, within the rough endoplasmic reticulum, ribosomes are often present as polysomes. There is no one ribosomes attached. There are multiple ribosomes attached in the membranes of the rough endoplasmic reticulum, and these ribosomes are attached in such a way that the mRNA is also attached with multiple ribosomes, and the ribosomes are attached with the endoplasmic reticulum and form as polysomes. So, what are polysomes actually? Polysome is a cluster of ribosomes. Okay, it is a cluster of ribosomes. That means many ribosomes held together by a strand of mRNA, which each transferring to translating to form a protein. So it simply means there are many ribosomes attached together and held with the mRNA, and with that same mRNA, they are using that same mRNA as a template for synthesizing proteins through a process called translation. And that protein is going to be inserted into the lumen of the endoplasmic reticulum, where they will be finally stabilized by post translational modification. Glycosylated, like for example, glycosylation occurs, and they are transported towards the Golgi apparatus for further stability, for further processing, so that they can be easily secreted out from the cell. Okay, so and these ribosomes actually found in this way. So if I see the previous diagram here, okay, I am watching this diagram now. So the pink ones are the ribosomes composed of large and small ribosomal subunit. Okay, and I have to draw thin line. So suppose this is the mRNA that you can see. This is the mRNA that you can see, and within the same mRNA, okay, a single strand of mRNA held together by multiple ribosomes, 5 prime, 3 prime. It is held by the many ribosomes are held with that same mRNA to form proteins. Well, the proteins, I am drawing the proteins with black color so that you can understand. The proteins are going to be inserted inside the cell, inserted inside the cell. Okay, and then they are packaged, they are further modified for their stability in their structure. Okay, like glycosylation occurs and different type of other events occur for maintain their structure. Okay, because these proteins are further they are linear in structure, so they has to be folded into their secondary conformation, tertiary conformation. Okay, so in this way they form the polysomes. So normally what it says, uh, that I have said before just now, that in rough endoplasmic reticulum, ribosomes are often present as polysomes, which are held together by mRNA and arranged in type of rosettes or spirals. Now, this rough endoplasmic reticulum attached with the mRNA. Okay, but how the the ribosomes attach with the mRNA? So, how the ribosomes attach to the endoplasmic reticulum to form the rough endoplasmic reticulum? Well, there are two transmembrane proteins. There are two transmembrane proteins called riboforin 1 and riboforin 2. These transmembrane proteins called riboforin 1 and riboforin 2 attach the ribosomes by their large subunit, which is the 60S subunit. Okay, so uh, so the ribosomes attach with the endoplasmic reticulum. The ribosomes are attached to the endoplasmic reticulum with the help of transmembrane proteins called riboforins 1 and 2. Now, if I say where it attach, well, ribosomes, I think you have studied in your junior classes a little bit in your class 12. The ribosome consists of two subunit. One is called the large subunit, one is called the small subunit. Okay, the large subunit is called the 60S subunit, whereas the small subunit is called 40S subunit. If this is your endoplasmic reticulum membrane that I am showing in pink, the large subunit attached with the large subunit of the ribosome attached with the endoplasmic reticulum with the help of a transmembrane protein, with the help of a transmembrane protein, and the name of the transmembrane protein is riboforin. Well, I am not drawing the accurate structure of riboforin, but I am showing that it is a transmembrane protein. It is found on the hydrophilic side as well as on the hydrophobic side of the ER membrane, where the protein synthesis is being occurred by using the mRNA as a template. Okay, so I have tried to draw here, but I think you cannot able to see it very clearly because it is already getting pixelated. These are the large and small ribosomal subunits. Well, this is the mRNA, and this is your riboforin, which I will show you in green. These are the riboforins where it attach with the large subunit and the proteins to be inserted, allowing the proteins to be inserted into the lumen of the plasma membrane, into the lumen of the ER membrane. Okay, these are actually the membranes of ER, of RER, better to say, rough endoplasmic reticulum. So now about the enzymes about the enzymes of your ER membrane. Let us see. There are various enzymes which are found within the membranes of endoplasmic reticulum, and some of the enzymes are found on outer surface of the membrane of ER. Some are found inside the lumen, okay, in the cytoplasmic in the luminal phase of the plasma membrane. So what it says, let us see. So the membranes of the endoplasmic reticulum are found to contain many kinds of enzymes which are needed for various important synthetic activities. So if you see the membranes of the endoplasmic reticulum, they contain many types of enzymes. Different enzymes are found which are needed for various important synthetic activities. And some of the most common enzymes which are found to have different transverse distribution of the ER membrane has been shown in the figure 6.1. It means that these enzymes are found on different sides of ER membrane. Some of the enzymes are found on the cytoplasmic phase of the plasma membrane of the ER membrane. Some of the enzymes are found on the lumen, that means inside the hollow space of the ER membrane. And the most important enzymes which are found on the endoplasmic reticulum membrane are stearases, NADH cytochrome C reductase, NADH diaphorase, glucose 6 phosphatase, and magnesium ion activated ATPase. Again, I repeat, these enzymes are found on the membranes of endoplasmic reticulum, and these enzymes are distributed on endoplasmic reticulum either on the cytoplasmic side phase, on the cytoplasmic phase, as well as on the luminal phase. Okay, okay. Now, and they are important. These enzymes are very important because they perform various synthetic activities. And the most important enzymes which are found on the endoplasmic reticulum membrane are stearase, NADH cytochrome C reductase, NADH diaphorase, then glucose 6 phosphatase, and magnesium activated ATPase. Well, if you see this enzyme glucose 6 phosphatase, these glucose 6 phosphatase are actually found on the luminal surface of the plasma of the ER membrane. Okay, luminal phase means that means the membrane where glucose 6 phosphatase are observed, they are found on the luminal side in the hollow space region of the ER membrane. Okay, other enzymes are also there, like your like for example magnesium activated ATPase. Magnesium activated ATPase found on the cytoplasmic phase of the ER membrane. Okay, it is found on the cytoplasmic phase of the endoplasmic reticulum membrane. Okay, now certain enzymes of the endoplasmic reticulum like nucleotide diphosphate is nucleotide diphosphatase are involved in the biosynthesis of phospholipid, ascorbic acid, glucuronide, steroids, and hexose metabolism. So there are certain enzymes of the endoplasmic reticulum like nucleotide uh, phosphatase. nucleoside diphosphatase. It will be nucleoside diphosphatase. They have said nucleotide diphosphate. It is not nucleotide diphosphate. It will be nucleoside diphosphatase. Diphosphatase. This is given on your table 6.1 right after this paragraph. So nucleoside diphosphatase are involved in biosynthesis of your phospholipid, ascorbic acid, glucuronide, steroids, and hexose metabolism. Okay, now all these enzymes, if I say together, that are found on the endoplasmic reticulum, perform the following important functions: synthesis of glycerides like triglycerides, phospholipids, glycolipids, and plasmalogens are synthesized by these enzymes. Metabolism of plasmalogens, synthesis of the fatty acids, biosynthesis of the steroids like cholesterol biosynthesis, steroid hydrogenation of unsaturated bonds. Okay, then other functions like NADPH2 plus O2, which is requiring for steroid transformation. Okay, NADPH2 plus O2 requiring for steroid transformation like aromatization and hydroxylation, side chain oxidation, deamination, thioester oxidation, desulfuration. So these are required for steroid transformation, and for this we require this one, NADPH and O2. Then ascorbic acid synthesis, as I have said, that this nucleoside, um, just now I have said, na? Okay, nucleoside diphosphate. It will be nucleoside diphosphatase. The name has been written wrong here. Nucleoside diphosphatase. It is also involved in ascorbic acid synthesis. Then UDP, uronic acid metabolism, UDP glucose dephosphorylation, and aryl and steroid sulfatase. So different types of functions are being done by these by these enzymes, different enzymes which are found on the endoplasmic reticulum. Okay, so you have to study this one, that if they directly give you a question that what are the main functions of the enzymes found on the ER membrane, including if you want to discuss about the direct functions of the enzymes, a direct function of the endoplasmic reticulum, then you can also give you this important point, that endoplasmic reticulum, its membrane consists of various enzymes which are involved in these synthesis, and the important enzymes which are found are this one. Okay, this one. So this is the table that I was talking about, that within this table it shows that where these enzymes are located on the membrane. Like you can see that NADH cytochrome B1, NADH cytochrome B5 reductase, they are found on the cytoplasmic phase of the membrane, whereas you can see ATPase, magnesium dependent ATPase, they are also found on the cytoplasmic phase. Well, one thing that I have not said, okay, glucose 6 phosphatase. Glucose 6 phosphatase is found on the luminal phase, that means on the empty space of the membrane. Okay, then beta glucuronidase, they are also found on the luminal phase of the membrane. Well, this one that I have said just now, nucleoside diphosphatase, which is involved in nucleoside diphosphatase is involved in biosynthesis of phospholipid, ascorbic acid, glucuronide, steroids, and hexose metabolism. This nucleoside diphosphatase is also found on the luminal phase of the membrane. Cytochrome B3, sorry, cytochrome B5. This enzyme is needed for synthesis of unsaturated fatty acid. They are found on the cytoplasmic phase of the ER membrane. So this was this was all about your enzymes of the ER membrane. So up to this, I completed. I want to finish my lecture. In the next class, I will discuss about the functions of the endoplasmic reticulum. There are some common functions which are found in both rough and smooth endoplasmic reticulum, as well as there are some separate functions as well. So here you can see the functions of the smooth and the function of the rough endoplasmic reticulum. See how steroid synthesis occur. How steroid synthesis occur in the smooth endoplasmic reticulum. You can see here. Then function of the rough endoplasmic reticulum, it shows that how the proteins are going to be inserted into the lumen of the endoplasmic reticulum. This is the lumen as written here. This is the cytosolic side, and these are the ribosomes. Okay, well, I will give you a animation. I will give you a video also, video animation on how the proteins are going to be transported into the endoplasmic reticulum lumen after protein synthesis. Okay, so just after when I will say study this, I will teach you this video lecture. The next video lecture, I will also send you that video, or maybe today also I can send you the video, so that it will be easy for you to remember. Okay, so this was all what we have to study for the ER. Not all of them, I will say part by part. So if you have any doubts regarding this lecture, you can ask me in the WhatsApp group.